Okay, thank you very much. So our last speaker of the morning session is Andrew Phipps from Sequela. And welcome, Andrew.
Thank you. Okay, so I'm Andrew Phipps from Sequela, and I'm going to give an update on two drugs in our pipeline, sutazolid and SQ109. I'd like to start out with some acknowledgments. I'd like to acknowledge NIH, the Division of Microbiology and Infectious Diseases, and the Division of AIDS for their support. I'd also like to mention the investigators of the Pan-African Consortium for the Evaluation of Anti-Tuberculosis Antibiotics. And I'll talk a little bit about the MAMS-TB01 study with SQ109. And I'd like to thank the organizers of the workshop for inviting us to talk about sutazolid and SQ109. So most of you know sutazolid is an oxazolidinone, a protein synthesis inhibitor. We licensed sutazolid from Pfizer in 2013 for worldwide commercialization on all indications. Sutazolid has excellent antimicrobial activity against Mycobacterium tuberculosis, and phase 1 and phase 2A safety studies were completed, and the early bactericidal activity is shown in phase 2A, and I believe the speaker earlier also talked about that very briefly. Sutazolid works well in combination with SQ109 as a companion drug, and we've looked at that in vitro and in vivo. So an update on the development of sutazolid. We're in the middle of a 6-month repeated oral dose toxicity study in rats that was started in January of 2016. That should be completed by October of 2016. We're also working with DAIDS to support tablet formulation of a 600 milligram API tablet, which we hope will be completed in 2016, and also manufacture of 600 milligram tablets in the end of 2016, early 2017 to support future clinical studies. We're also working with them to manufacture PNU 101-603, which is the major active metabolite of sutazolid. And this will be used for reference material, but also to support bioanalytical studies, mainly hollow fiber studies, looking at the activity of sutazolid in combination with its active metabolite. So I'd like to focus more on the development of SQ109. SQ109 is an ethylene diamine drug. There have been multiple mechanisms of action that have been mentioned or identified to date. Originally, SQ109 was targeting MMPL3, which results in the accumulation of trehalose monomycolate. We've also identified that SQ109 targets MEN-A and MEN-G, menaquinone A and menaquinone G, which affects respiration and electron transfer. SQ109 also collapses proton gradients, so proton motive force, and both of these lead to decreased ATP biosynthesis. And because of these two, the decrease in ATP biosynthesis, we see a reduction in ATP binding cassette transporters. And so combined together, we have multiple mechanisms of action that have been identified for SQ109. So looking at Mycobacterium tuberculosis, we have MMPL3-related mechanisms of action and also a decrease in ABC transporters. And SQ109 has activity against other bacteria, Helicobacter pylori, fungi, Candida albicans, and Trypanosoma cruzi, all which lack MMPL3. So to date, there have been no severe adverse events related to SQ109. We have seen some hepatic toxicity in SQ109 containing arms with drugs known to be associated with hepatic toxicity from the MAMS-TB study. SQ109 has been used in subjects with drug-sensitive TB and MDR-TB. There have been over 260 subjects that have received SQ109 at doses ranging from 75 to 300 milligrams per person per day, and the duration has ranged from 2 weeks to 6 months. We are working with DMID to analyze the SQ109 plasma concentration in samples from the MAMS-TB01 study, and we hope to have that completed by the end of 2016. Sequela is partnered with the Maxwell Biotech Group, and there has been a clinical trial of SQ109 conducted in MDR-TB patients in Russia, and The last patient, last visit has occurred, and the data analysis is ongoing, and we expect to have a submission of a new drug application to the Russian regulatory agency sometime in 2016. So I wanted to talk a little bit about SQ109, and just to remind you of the clinical trial design here. The control was isoniazid, rifampicin, pyrazinamide, and ethambutol for 8 weeks, followed by a continuation of 12 weeks of isoniazid and rifampin. The SQ109 containing arms were isoniazid, a standard dose of rifampicin, pyrazinamide, and SQ109 for 12 weeks, and then there was a higher dose of rifampicin at 20 milligrams per kilogram, isoniazid, pyrazinamide, and SQ109. I won't be talking much about the moxifloxacin containing arm or the high-dose rifampicin at 35 milligrams per kilogram. But what I wanted to talk about was really looking at sputum culture conversion. And so what I have here is the data showing the percentage of negative sputum cultures taken at various time points in liquid and solid media. And I want to focus mainly on the difference between the standard of care at 12 weeks. So the original study design was to look at the time required for a sustained sputum culture conversion. And while we understand and support definitely looking at regimens that can shorten the time required, I'm not sure that this paradigm is going to fit with all drugs. And so to look at this really at the percentage of sputum culture conversion, we can see at 12 weeks on liquid culture that the arms containing SQ109 basically were equivalent to the standard of care in terms of the percentage of subjects that had a negative sputum culture at 12 weeks, which ranged from a 2% decrease to a 2% improvement over the standard of care. But if you look at the 95% confidence intervals, they completely overlap. So I wanted to talk a little bit about sputum culture conversion and using that, and I think that we discussed a little bit of this yesterday, that when we look at decreasing bacterial burden in sputum, that these two diagnostics probably have a different lower limit of detection. So when we look at a different lower limit of detection, we actually see a different time required to reach a sustained sputum culture conversion. But I think that we would all agree in the early part of therapy that bacterial burden is decreasing, and that's reflected in what's happening in sputum. And when we reach the point at which we've gone below the limit of detection in liquid, this becomes the black box, that at this point in the trial, we really can't tell what's happening other than that the sputum culture is negative. And so I've shown here the 8-week percentages in both solid and liquid, along with the 95% confidence intervals, and the band across in gray. And with the SQ109 containing arms, essentially we're within the confidence interval of the standard of care in liquid and maybe slightly above in solid. However, I think what's still remains to be determined is what's happening when we can no longer pick up colonies or a positive culture in liquid from the time that we continue treating until we actually finish treating, and then what determines really the difference between a favorable outcome and relapse. Essentially, we have to monitor the subjects and then look at the number of the ones that relapse. So the assumption is that shorter time to conversion, see if I can point here without messing this up, would result in a shorter time to whatever happens in this black box, which would result in shortening the entire length of treatment. So basically looking, I can't point over there, but looking at this decrease in time. However, we don't really know that. And without tools like imaging or other ways to examine what's happening here, we're left with, what are these drugs doing in this period of time that actually results in a favorable outcome? So time to sustain sputum culture conversion tends to favor drugs with a more rapid bactericidal response, which we know may not be the case with SQ109. And that's not the case with some of the other TB drugs. And that using this, while we can try to determine if we can develop regimens that can shorten therapy, it may not actually be appropriate for all of the drugs we're trying to develop clinically. So what are the activities of drugs required for preventing relapse? And unfortunately here we don't really have the tools to measure this right now. So if we continue on looking at 12 weeks and 26 weeks, essentially there's minimal differences in the proportion of subjects with a negative sputum culture conversion compared to the standard of care at 12 and 26 weeks. and the substitution of SQ109 for ethambutol in this trial, or moxifloxacin for ethambutol in a regimen containing isoniazid, rifampicin, and pyrazinamide is essentially equivalent to the standard of care when we look at an outcome that's not based on the time for sustained sputum culture conversion. The other thing is if we look at the proportion of patients who were able to achieve a favorable outcome at 22 and 26 weeks is also comparable to the standard of care in the SQ109 containing arms, and that there were no subjects that actually failed according to the definition, which is a positive culture at 22 and 26 weeks, and that when you look at the favorable outcome, 63% versus 73% versus 76% for the control group, that subjects in the SQ109 arms were able to achieve a favorable outcome at 26 weeks in this trial. So in summary, I'd just like to say that SQ109 is well tolerated and has a favorable safety profile. We've talked about that it's been in over 300 subjects so far with drug-sensitive and multi-drug resistant TB with no severe adverse events reported. We need to do more with the pharmacogenomics. We believe that polymorphisms in CYP2D6 can be related to differences in exposure between individuals, and that we need to understand the contribution of CYP2D6 and 2C19 to SQ109 metabolism. We need to further investigate the potential for clinically relevant drug-drug interactions. We have some concerns over using SQ109 in combination with rifampicin, especially in individuals who have reduced metabolism due to 2D6, in which induction of 2C19 may reduce their exposure. And we also need to establish PK/PD relationships for SQ109, and we also need to evaluate an appropriate human dose based on systemic exposure. So we've gone as high as 300 milligrams per person per day. We're not convinced that that is the most or the optimal dose. I think that without having a good understanding of the PK/PD relationship and being able to look at systemic exposure and outcome, we need to be able to evaluate, do we need to go higher on the human dose in order to have a maximal efficacy? But we also need to understand that the potential for drug-drug interactions when SQ109 is combined with other antimicrobials to make sure that we actually aren't altering the exposure of SQ109. And that's essentially a very quick overview from Sequela. I'd be happy to answer any questions or comments.
Thank you, Andrew. Any questions just for clarification?
This is Erica Lesson from Treatment Action Group again, and a question for clarification would be, on what grounds is this drug getting registered in Russia, and will the data actually be made available, because it doesn't seem like there's sufficient to warrant registration. And then second, on the sutazolid front, I guess it's a comment, but I think it's really a tragedy that the drug was licensed to you guys since 2013 and absolutely nothing has happened with it in terms of clinical studies, when we've seen here today that there have been huge needs for more oxazolidinones to be in research.
Thank you for your comment. I'm willing to address the comment concerning SQ109 and registration in Russia. So we've been working with the partnership, the Maxwell Biotech, to evaluate SQ109 clinically. There was a study done in MDR-TB that is comparable to a phase 2b study conducted with other drugs, including bedaquiline and dilamidid. So this would be 6 months of therapy in OBR plus SQ109 versus OBR and placebo for 6 months, followed with a continuation phase. I didn't go over all of the non-clinical safety pharmacology with SQ109, but the Russian regulatory agency has accepted the non-clinical safety pharmacology studies that have been done to date, in addition to the phase 1 studies and the phase 2A studies that have been conducted in SQ109 to move forward with registration, based on a conditional marketing authorization at the conclusion of a phase 2B study.
Okay, Andrew, thank you. Oh, sorry.
Thanks, Andrew. Maybe just one brief clarification on the MAMS study and the outcomes that were presented. So as you alluded to the time to negativity outcome being in debate over whether it's really associated to a long-term benefit, we had just taken an early look on the association of this outcome to the long-term outcome that patients were having in the study, and it seems that actually the time to event outcome, especially in liquid media, correlated very well with the long-term outcome of relapse or recurrence at this point versus no recurrence. So just to add that to the focus on individual time points that you've presented.
Thank you.